In Spain, we have a saying, I'm going to translate, that says that if you strike first, you strike twice. In that regard, we are going to listen talk about early diagnosis of cancer. And it's Xiaojun Chen, who's also an associate professor at the College of Chemistry and Molecular Engineering, who also happens to be the deputy dean. And she works at the College of Chemistry of Nanjing University of Technology, and she was a visiting scholar at Oxford University, and she has published several papers and also tutorized several PhD works, and is the lead of up to 6 projects, including National Natural Fund, JAF Fund, and Jiangsu Provincial Natural Fund, with up to 1.2 million yuan in funding. Thank you, Xiaojun.
Thank you. Good morning, everyone. I'm so happy to stand here to communicate some scientific research with my friends in Spain. And thanks for the invitation for the government of Aragon and the University of Zaragoza. Today, my topic is electrochemical diagnosis of lung cancer based on different biomarkers. I come from Nanjing Tech University. You see, the tumor marker. The tumor marker is the specific signal for the diagnostic of tumor. And the tumor marker, the types, including such as antibody, protein, RNA gene, including mRNA, microRNA, link RNA, etc. And you see from this the second figure, you can see that in the clinical diagnostic, the tumor mass is sometimes excess 1 gram. At this time, maybe the tumor in the middle or the late period, and it is hard to save one person's life. And in the biosensing field, the early diagnostic, using the tumor marker, at this period, the tumor mass is about 1 milligram, and at this time, maybe the tumor just on the stage of the early period. Maybe we can use the sensitive detection method to save more people's life. Now, the detection of tumor marker, the method including immunoassay, tomography, endoscope, biochips, cytography, and biosensor. Among this method, the biosensor may be the sensitive, simple, rapid, and maybe inside detection. Now we need some signal amplification strategy in biosensors to get the high sensitivity. Normally, the method may be lying 3 aspects. The first one is surface modification, the second one is nano labels, and the third one is using the cyclic strategy. The first one and the second one using the nanotechnology to help us to get the aim. The surface modification in our group these years using the nanomaterials such as the 3D array, including the 3D nanoparticles array, 3D macroporous array, 3D organic and inorganic array, and sometimes the self-assemble of alloy gold array, The nanolabels in our group, including the nanoparticles, the nanotubes, nanofibers, hollow nanoparticles to embedded or to modification for the for different molecules, and using the various enzymes such as the DNA enzyme and the polymerase and the nicking indicase to help us to complete the cyclic strategy. And this year, we also publish these papers already. Today, I will introduce to you several of our representative work. The first one is the EGFR immunosensor based on 3-DOM. 3-DOM, that is the 3D order macroporous gold nanoparticles, polyethylene 3 acetic acid. Here, I can introduce to you the normal strategy to produce the 3-DOM structure. We usually use the nanoparticles self-assembly on the surface of the electrode, then electrodepleted or the physical absorption, the material to depletion into the interspaces of the nanoparticles. The nanoparticles normally using the silica particles or the polystyrene particles. After the electrodepletion, then we can use the hydrofluoric or the hydrofluoric to dissolve the silica template, and or using the THF to dissolve the polystyrene template to get the 3-DOM structure. Then we immobilize the antibody onto the surface of the electrode, and BSA block the remaining active sites. In the presence of the target, then the secondary antibody could immobilize onto the electrode surface. Then usually the secondary antibody need to be labeled with the nanoparticles to increase the immobilization amount. Here we use the gold nanorod as the nanolabels because it can catalyze some small molecules. Here the gold nanorods can catalyze the reduction of hydroperoxide, and in the presence of TMB or MB, methylene blue, such as the electrochemical probe, then it can give us the electrochemical signal. Here some characterization. The first one is this 3-DOM structure of PTAA film. And the second one is the gold nanorod. Then you can see from this figure, along with the EGFR, the target concentration increase. Then we can increase the current signal of MB, methylene blue, according to the electrocatalysis between the gold nanorod with hydroperoxide. And we can get the linear range, and we can see in the lower concentration range the sensitivity seems larger than that of at the bigger concentration range. And the second work is targeted triggered triple isothermal cascade amplification strategy for ultrasensitive microRNA 21 detection at sub-attomolar level. In this work, the target triggered triple, that is 3T isothermal cascade amplification, 3TICA strategy for the ultrasensitive microRNA 21 detection. In this work, we can see from this scheme 3 gears they work successively. You can see that the most important step is the first cycle. In the presence of the target, and here we use the microRNA 21 as a model. Actually, you can use many targets to replicate this cycle. In the presence of the target, we design this template strain. Here, in the template strain, we have three main parts, A, and B. In the presence of T, the T is complementary with the strain A, and with the aid of polymerase, the strain can be formed, and after nicking, the upper strain can cut it into three parts. The first one is a T. The second one is a T estron. That means this part, T estron, the difference between T and T estron, that is T is the strain of RNA, and the T estron, that is the strain of DNA. And after the cycle one, small amount of T can cause a large amount of T estron, then cause the amplification of this part of the complementary strain of B. Then we call this C strain. The C strain is an DNA containing strain. You can see in the cycle 2, the C strain can complementary with partially complementary with the substrain. In the presence of zinc ion, the RA base can cut into two spaces, two parts, and here we can got the purple strain, and in the presence of heming, it can fold it into a G quadruplex structure. It can catalyze the reduction of hydroperoxide, and in the presence of TMB, that is another electrochemical signal molecule can get the electrochemical signal. And here you can see small amount of T can get large amount of G quadruplex structure, and we can got the amplified electrochemical signal. Here in this work, we get an important parameter, that is the length of C, and that is the design of C strain. C strain, you can see from this table, we have C16, 20, 24, 28. And the difference between them, you can see from the origin basis. The difference is the length of the complementary part of C strain with the substrain. Then we think if the complementary part is too short, maybe it cannot obtain the stable hybrid, and if they are too long, in the presence of zinc ion, the RA cut it, and otherwise both this part, the two parts could not break separately. So we can we should optimize the length of the C part. And in the in the conclusion of our experiment, we can see that C24 is the optimized length. It can satisfy the stable structure of C with sub, and in the presence of zinc ion, it can separate rapidly. And from this figure, we can see the linear range of microRNA. The detection limit is about 1 attomolar, and the linear range from 1 attomolar to 100 picomolar, several ranges. Okay, the second work is a novel long-coding RNA electrochemical gene sensor based on three-dimensional ordered macroporous gold nanoparticles, reduced graphene oxide composite. The similar way to get this 3-DOM structure, and this time we use the graphene oxide and gold nanoparticles co-depleted into the interspaces of the polystyrene template. And in the presence of the specific sequence of non-coding RNA, and we can get the electrochemical signal from the probe of RUHEX. And here some characterization. You can see that we prepared successively the 3-DOM electrode interface and the nano label, and we also characterize the hybridization with the target and DNA1, DNA2. We still get the linear range from 1 picomolar to femtomolar to 50 nanomolar. The fourth work is our recent work, double determination of non-coding RNAs from non-cancer via multi-amplified electrochemical genosensor at subfamilial level. Here, we detected the MEX3 link RNA from non-cancer using two routes. You can see from here we just have two targets. The two targets is from the specific sequences from the MEX3 is selected from the MEX3. We use two main amplified strategies. The first one is the magnetic nanobeads aided recycle, and the second one is the HCR reaction, and we get two electrochemical signals of methylene blue and the ferrocene as the probe to get two separate electrochemical signal. We can got the some characterization about our interface using the XRD, TM, SEM, etc. And we can prove the effective of our strategy, especially from this figure. You can see that we use the HCR strategy can cause the signal increased about 4 times. The linear range for both two targets from 1 femtomolar to 100 picomolar, and we verified its specificity using different specific chains from other linked RNA. And the last one is our recent work, latest work, a dual channel detection of hotel. Another non-cancer related linked RNA using padlock probe based exponential rolling cycle amplification assay. And in this work, you can see that we use both the electrochemical and the illuminance to fabricate the signal off and on mode to detect two targets simultaneously, and we use the fluorescence probe and the electrochemical probe, using two ions use the DPV method and the fluorescence method to get the dual signal to verify each other. Okay, then at last, I'd like to thank all my students and all my foundation to support my research, and thank you for your attention. Thank you.
Thank you, Xiaojun.